All right, so right now we're going to look at carbonic anhydrase. So carbonic anhydrase is this metalloenzyme that is plays a big role in maintenance of acid-base balances and as well as working with the transport of carbon dioxide to be released outside of the body. There's three different types. There's alpha, beta, and gamma subunits. In the animals, it's majority of the carbonic anhydrase is in the alpha configuration. And so this main function is conversion of carbon dioxide into a sense of bicarbonate. All right. So the most well-studied carbonic anhydrase is carbonic anhydrase 2, and that's the specific type of enzyme that's found in red blood cells, right? And so the main function is, when we look at these different transports, is the conversion of carbon dioxide, CO2, into bicarbonate, right? So we have carbon dioxide and it needs to be removed from the tissues. So you have carbon dioxide combining with water, gives you this reaction. It is a reversible reaction, which gives you carbonic acid, H2CO3, right? But because an acid, definition of acid is what? Is a proton donor, this acid actually dissociates, and your remaining byproduct is a hydrogen ion plus a bicarbonate, HCO3 minus, right? And so this hydrogen group serves as an acidic function, and this bicarbonate plays a big role as a base, right? It stores in the pancreas, where it's released, for example, instances of during digestion, whenever the acid, the chyme from the acid of the stomach gets released into the intestine, this bicarbonate is released into the duodenum of the small intestine in order to neutralize the acid, right? And so this carbonic acid plays a big role here, specifically as the conversion of, let's use pink. Carbonic acid here, where it converts the carbon dioxide into water, the carbon dioxide and water into carbonic acid, right, which eventually dissociates because this acid is specifically in a polar medium, right, in water. It donates its hydrogen, right, into the medium, and you are left with a bicarbonate that's present, right? So this entire process is happening in the red blood cells specifically, right? And the whole function of this enzyme is to decrease the activation energy in order to speed up the reaction, right? So carbonic anhydrase is an enzyme, right? So the main function of an enzyme is a catalyst to speed up the reaction, whether it's forward or reverse.